Alright, in this video we are going to showcase one of the microorganism, which is Pseudomonas aeruginosa. So this selected bacteria, which is highlighted in the red, they are all gram negative. So when you're coming in the lab, you're going to experience gram staining. Just be aware that they are gram negative. And then now we are going to actually cover this box, and Sanjida is going to demonstrate with the oxidase and OF medium and motility. So let's go to the other side where Sanjida is going to work.
Hello.
In aseptic condition. So what we are going to do, Sanjida?
So the first step is definitely we sterilizing the loop, and the organism we'll be using it's Pseudomonas aeruginosa. So after sterilizing my loop, I wait for 10 to 12 seconds, and then from the culture plate I get some culture to do the oxidase test. And the way I'll be doing it, so these are the oxidase strip. So as you could see, that's how the oxidase strips look like. So now once I place the sample on the strips on this side,
then they can see the result straight away. So if
as you
after
could see, this has changed the color to blue. That's an indication of the organism being oxidase
positive. When students come to the lab, they have an experiment. They work with the oxidase, which is the same stick, and that is not going to be turned blue, and that is oxidase negative. But for Pseudomonas, it's oxidase positive. And in fact, all of the bacteria for this session, they're all oxidase positive. All right.
The next section I'll be doing with OF tubes. So I'll be using this long wire for these OF tubes. So what I will do, I will definitely sterilize the loop first. And then from the plate culture, I take the sample.
So we work with one sample, but you are going to observe the results for all the 4 selected gram negative, oxidase positive.
So what I'm going to do, I'm stabbing this wire inside this medium all the way down, and I just take it out. And I do the same for the other OF tubes.
And the idea here is you're going to add the sample to both of these OF tube, and one of them is going to be covered with the
Paraffin
oil to make it anaerobic condition. So your observation for your selected bacteria is to see how they are going to react and grow in OF tests, OF tube without oil and one with oil.
So what I'm going to do, I'm going to add 1.5 ml of this paraffin oil in one of these OF tubes. And we will see the results after incubating at 30 degrees for 2 days.
Right.
And then
the last part
will be the motility test that we use using this loop. So definitely the first part will be sterilizing the loop, and in this section we will be using either broth or plate. So I will just demonstrate using the broth.
So students already experienced in the previous prac working with the loopful of the broth culture, right, one drop in the middle of the motility plate. And I guess this is the end of this video.
As I said, we will be just placing one drop in the center of this plate, and we will see if this organism is motile or not after 2 days of incubation at 30 degrees.
Thank you, Sanjida.